Dear colleagues, the previous speaker told the audience about the clinical value of quantitative MRI based on magnetic susceptibility and pointed out what tissue characteristics it is associated with. However, what allows us to be sure that a new magnetic resonance biomarker reflects certain specific tissue properties or disease-related tissue changes and, moreover, is suitable for their quantification. Obviously, the criterion for the clinical value of MRI biomarker can be histology, which is the gold standard for validation of imaging experiments. The scheme on the slide shows the place of histological validation in the process of a new imaging technology development. New MRI technology develops to improve diagnostics and based on clinical data and physical theory. However, rigorous histological validation is required to assess current and potential value of new MRI technique. For this, as a rule, special studies are carried out in animal models under controlled conditions, but histopathological studies of human postmortem tissues are also important. The strong correlation between MRI and histology, confirmed by many studies, opens the possibility of using this technology in clinical practice. If the correlation is not confirmed, optimization of MRI technology and new validation studies are required. So, histological validation is the most important evidence that proves the reliability of specific biomarker in preclinical and clinical applications. Any validation study generally includes several steps. Selection of animal model or human samples for research. Those can include normal brain, specific models of human disease, genetic models, neurodevelopment, and so on. MRI in animals or in human postmortem. Histological tissue processing. Measurements of MRI and histological images, and statistical comparison of MRI and histological measurements. Each of these steps is responsible and requires careful consideration. In my lecture, I will focus on histological processing and the correspondence between MRI and histological measurements. The first step for histological validation is the selection of animal model. This slide represents only a small part of widely used models of neurological diseases. These models vary in complexity of implementation, accuracy in reproduction of human disease, and suitability of species for a particular model. Among this variety, the choice of the most suitable model is quite difficult. I can only give a few recommendations for choosing an animal model. The first advice: the purpose of histological validation is different from the study of disease mechanisms. Therefore, the control of change in the biomarker is more important than the exact reproduction of human disease. Ideally, only one biomarker should change. Inflammation or gliosis can only complicate the task of histological validation, especially at the beginning of studies. For example, there are two well-known models of multiple sclerosis: the Cuprizon model and the model of experimental autoimmune encephalomyelitis, EAE model. Cuprizon induces demyelination with little or no inflammation, and therefore doesn't reproduce the signs of multiple sclerosis in humans. The EAE model is associated with an autoimmune component and causes inflammation similar to multiple sclerosis in humans. Despite this, the purpose of histological validation of myelin, the Cuprizon model is better because it allows to evaluate demyelination almost in its pure form. The second advice: carefully check the sensitivity of animal species for the model. For example, the coprazone model reproduces well in mice, while guinea pigs and rats are more sensible in the EAE model. And the third advice: assess the technical complexity and standardization of the model. A well-known model will give you more information from other studies and raise fewer questions when you will publish your work. The complexity of the model is not always an advantage. Evaluate the possibilities of implementation in your laboratory. And the first simple test question: which animal model would you choose for the histological validation of the MR biomarker? And this variance. The first choice: the model accurately reproduces the disease in humans, but causes many tissue changes, in addition to changes in the biomarker being studied, for example, inflammation. The first choice is incorrect. Validation is more reliable when only the biomarker under investigation is changed. The second choice: the model reproduces only a part of the signs of human disease, but the studied biomarker changes to a greater extent. The second choice is correct. Validation is more reliable when only the biomarker under investigation is changed. The next important step of histological validation studies is the pre-processing. Here are the features of two main types of pre-processing in animal studies: paraffin and frozen sections. The third type, fresh sections without fixation, are used only in special cases. For both types of sample preparation, fixation by transcardial perfusion with paraformaldehyde solution is preferred. This allows to make fixation more uniformly and avoid autofluorescence during immunohistochemical labeling. In our experience, some proteins can be detected without this procedure. Paraffin sections are thinner, stain faster, and can be stored at room temperature. The storage of frozen sections requires additional freezing equipment, and even a short defrosting is critical to sample quality. Despite of differences in preprocessing, both histological and immunohistochemical studies can be performed on paraffin or frozen sections. However, fewer antigens can be detected on paraffin sections than on frozen sections by immunohistochemistry. We choose frozen sections in our studies in our laboratory. Several features of histology of human postmortem tissues should be noted. Unlike animal studies, human tissues can be only collected naturalistically, and both pre- and post-mortem conditions may influence the accuracy of the results. A key factor in conducting such study is the quality of the tissue. To date, it has been proposed several markers of human postmortem tissue quality. Right side of the slide. This is postmortem interval, very important. The manner of death, sudden versus prolonged death, and pH of tissue are also important. And there are specific markers of RNA quality. The next test question: which of the following features correspond to paraffin sections, which to frozen section, and which to both? Let's check in order. First feature: fixation by transcardial perfusion is better. And it's true for both types of sections. Fast and uniform fixation is best for paraffin and frozen sections. The second feature: Long-term storage. This is correct for paraffin sections. It can be stored for several years. The third feature. Slices are thicker. Frozen sections. Usually frozen sections are thicker than paraffin sections. Slices can be labeled immunochemically. Both paraffin sections and frozen sections can be stained immunochemically. And the last feature, more antigens can be detected. This is frozen sections. The tissues are fixed and sections are obtained. Now we need to stain our sections with the biomarker of interest. This slide summarized the advantages, green color, and limitations, red color, of histological and immunohistochemical, here more advanced immunofluorescent staining. As you can see, histological staining of sections takes less time and requires less resources. These sections can be stored longer and cause fewer questions about their quantification. At the same time, immunofluorescent labeling allows to identify specific proteins and introduce additional markers for identification of the cell type, cell nuclei, and cell environment. This image can be easily decomposed into color or pseudo-color channels in contrast to histological images. An example is shown at the bottom of the slide. This is an immunofluorescent micrograph of a bundle of myelinated axons in the Khodanovich of red, right bottom side of the slide. The colored channels represent myelin axons and DAPI-stained nuclei. However, sometimes histological staining gives more reliable results despite less specificity. I will show such an example later. This is a list of the most commonly used specific histological stains and antibodies for the detection of CNS cells and components. Of course, this list is much longer. Below are examples of stains, and as you can see, double immunolabeling allows to observe several components of several proteins in one micrograph. Myelin with axons, myelin with microglia, neurons, or accumulation ferritin or by macrophages. A little more about histological validation of myelin. Myelin is composed of a lipid bilayer and internal and external proteins associated with it. 1/3 of myelin is proteins. The main myelin proteins are myelin basic protein and proteolipid protein, which can be labeled immunochemically. The main component of myelin is lipids. Therefore, the sulfase blue is most commonly used histological staining of myelin. According to meta-analysis that includes 70 MRI myelin validation studies, the most commonly used techniques are LFB, MBP, PLP staining, and electron microscopy. Electron microscopy investigates the outer structure of myelin. However, unlike other techniques, it doesn't allow to study whole brain sections, and this makes it difficult to compare MRI and histology in specific locations. Quantitative estimates may depend on which component of myelin we stain and what structure of the brain we are investigating. This is an example of how histological staining gives more reliable results. The study performed on the Cuprison model showed that the dynamics of demyelination may vary. for white and gray matter for different types of brain tissue. The upper figure clearly shows that almost complete demyelination was observed in the corpus callosum in 5 weeks after starting cobrazone treatment, while in the cortex in 6 weeks. Besides, the dynamics of different myelin components during demyelination can also vary. The lower figure illustrates the difference in myelin scores when using MBP and PLP staining. Note that LFB staining is the lipid component, gives average results. Therefore, the choice of a marker is extremely important, and the use of multiple markers gives more accurate estimates. We stained sections and got micrographs. The next step is segmentation, selection of regions of interest for analysis. Good correspondence of MRI and histological images and determination of regions of interest is critically important for quantification. Although histology is the accepted gold standard for imaging experiments, it should be noted that postmortem tissues are usually deformed during sample preparation. Therefore, finding matching regions of interest on histological and MRI slices is not a trivial task. There are several approaches to solving this problem. Two most widely used and simple approaches are delineation of the brain structures or placing standard regions of interest on image. Each of these methods has advantages and limitations. The delineation can include the boundaries of the explored structure with outliers affecting the means, while mean in standard ROI can depend on its location within brain structures. A more sophisticated approach involves co-registration of MRI and histological images, right side of the slide. In this case, deformable or elastic registration leads to distortion of the original MRI or histological image, which also distorts the measurement result. In our recent study, we used an approach based on ROI transformation, registration of only the ROI from the source image to the target image, and this allowed us to avoid measurement distortion. A method of histology quantification is determined by the type of staining. The most commonly used method for quantification of LFB histological staining is the composition of RGB image into channels and measuring the optical density, the intensity of the red channel. This channel is most contrasting and corresponding to the stronger light absorption in this regional spectrum. For other histological stains, the image can be converted to grayscale for measurements. And unlike optical images, for fluorescent images, the composition of RGB image into channels is univocal. Two quantification methods can be used: measuring the fluorescent intensity and thresholding, followed by determining the percentage of antigen positive area. When measuring the fluorescent intensity, it is important to use the same microscope settings and avoid channel saturation when photographing. In addition, quantification needs to be calibrated using a standard non-fading image or fluorescent microspheres. It is easier to avoid incorrect estimates by using the threshold method. And threshold methods are also often used for quantification of electron microscopy images, for example, for myelin sheath thickness, myelin volume fraction, or the ratio are measured. In addition, cell density can be used as a quantification. For example, we use cell density of immature elegant erythrocytes to assess the association of macromolecular proton fraction with remyelination. And one more example, classification of tumor type using perfusion-weighted MRI and cell density. The next test question. Which of the following features correspond to histological staining, which to immunofluorescence? And the first feature, long-term storage without fading. Histological sections, it can be stored at room temperature. The second feature, the biomarker corresponds to only one color channel. Of course, immunofluorescence is one of the advantages of immunofluorescent staining. The threshold method is more common for immunofluorescence. The measurement of signal intensity is less reliable. Multi-labeling is possible for immunofluorescence. One label corresponds to one color channel. And quantification uses optical density. Histology. Histological sections are photographed in transmitted light. The last step is statistical analysis. The simplest and widely used model is linear regression with the calculation of Pearson's correlation coefficient. The left side of the slide shows the results of our study performed on the cuprizone model of demyelination, confirmed highly significant correlation between macromolecular proton fraction and LFB optical density using linear regression analysis. More complex models that uses two or more parameters to predict histological features can be represented by multiple regression. The middle part of the slide shows how including additional parameters to the statistical model can improve it. The study performed on the model of ischemic stroke, 10 days of post-stroke observations showed that percentage changes in LFB optical density and MPF were strongly correlated. The fewest correspondence was found for the one-day time point due to the presence of edema. Addition of T2 into multivariate regression further improved agreement between MPF and LFB optical density due to correction of the edema effect. The right side of the slide presents a new approach, validation using classification by deep neural network analysis. Hematoxylin eosin stained images were co-registered to MRI. Then expert assessment was used for neural network learning. Trained network allowed to differentiate 6 types of tumor with accuracy of 86%. And conclusion. Histological validation is the most important evidence that proves reliability of specific biomarker in clinical applications. Each component of validation study is important and affects the results. When choosing animal model, it should be considered that the control of biomarker change is more important for validation than the exact reproduction of human disease. Both histological and immunohistochemical studies can be performed on paraffin and frozen sections. However, most antigens are better detected on frozen sections. Immunofluorescent labeling has a high specificity and more extensive possibilities, but sometimes histological staining gives more reliable results. Image processing is determined by the objectives of the study and the type of staining. For optical images with histological and immunohistochemical staining, optical density is more commonly used. For immunofluorescent images, the threshold method is more commonly used. Statistical analysis can include both simple or multiple linear regression, as well as new approaches based on training neural networks. And my acknowledgements for the colleagues in Russia and in the United States. And I apologize that I cannot participate in the conference personally. And thank you so much for your attention.